Reader Digest magazine. First of all, thank you so much for making time for this interview.
You're welcome.
Our issue for this month is about how pharmacogenetics can treat acute lymphoblastic leukemia. So, doctor, can you explain to us more about this topic?
Okay, so first of all, to understand this topic, you need to know what pharmacogenetics is. So pharmacogenetics is the study of inherited genetic difference in the drug metabolic pathway in terms of therapeutic effect as well as adverse effect. So now let's talk about what is acute lymphoblastic leukemia. Acute lymphoblastic leukemia is a common malignancy in children. So it's a malignancy of the bone marrow where the blood is made. So this is caused by excess production of lymphoblasts, which are immature white blood cells.
Have you had any interesting experiences in handling this case?
Actually, I did have. You see, I had two cases. I had two patients in the past where their results were actually very interesting because they showed different reactions even though they were given the same drugs. You see, I had two patients. One was named Lily. She was very active, and since she was the lead of her soccer team. And another person was Sandhya. She was more involved in theatres and she led a sedentary lifestyle. But the thing is, both of them were sick and they were diagnosed with acute lymphoblastic leukemia. They did exhibit common symptoms, but the major difference between them was Lily had bruises. She had black and blue bruises all over her body, but Sandhya suffered from infections and fevers. But after some tests, they were both diagnosed with acute lymphoblastic leukemia. They were given the same dosage, and but they exhibited different reactions.
How can this happen?
So you see, they were both given thiopurine drug, which is also known as a 6-methyl-thiopurine drug. And when they become active, they become thioguanine nucleotide, which means they incorporate into DNA and RNA. They cause DNA protein crosslinks, single-strand breaks, inter-strand crosslinks, and sister chromosome exchange. They will disrupt replication and transcription process, which means rapidly dividing cells, such as cancer cells, will not survive. But the thing is, the downside of this drug is that it is not selective for cancer cells only, which means they can be toxic to our body.
So, how do you inactivate the drug then?
Okay. To inactivate this drug, there's this enzyme called thiopurine methyltransferase, TPMT, and they add methyl group to the 6-MP drug, and which means they prevent the drug from entering the active pathway. And since they will not be entering the active pathway, they will be cleared from the body. And you should know this, that TPMT's main function is to inactivate these drugs. So as you can imagine, if there's a deficiency of the TPMT drug, TPMT enzyme, there's a high conversion of the toxic TTNs. In my patient's case, after giving this thiopurine drug, Lily's condition worsened. Her vital signs plummeted, her anemia worsened, her erythrocyte count went down, and her heart function was compromised. Fevers were spiking, her breathing was shallow and labored. She wasn't eating properly, which means we had to hydrate her intravenously. Her condition was life-threatening. In contrast, Sandhya got better. Her anemia went down. She wasn't suffering from fevers. She showed signs of appetite and boredom, which means they are good indicators of improvement of health.
Oh, that is a shocker, doctor. What could possibly cause this condition? How could a drug possibly affect each person differently, and how did you manage to overcome this situation?
Well, you see, ma'am, the girl's reaction to the treatment was very different due to their genetic difference that alters metabolism of the chemotherapeutic agent. You see, different alleles of the TPMT gene lead to some alteration of protein function. In this context, each individual possesses either high or medium or low TPMT activity based on inheritance. You see, there was research that was conducted on the distribution of the TPMT activity in 298 Caucasian adults. You see, some had low, some had high, and the majority had medium TPMT activity. So we can conclude that people with low TPMT activity has high TGN level, which we can conclude that there's a high level of toxicity.
Since we were talking about genetic differences, can mutation play a part in a girl's different reaction to the drugs?
Well,